Hi guys, happy Wednesday. So today and tomorrow I want to kind of go through some review things. Tomorrow, Thursday, will kind of be a little bit of an off day for you, but I'll be available for questions. And Friday's your test for DNA and protein synthesis. So the first thing, if you have yet to turn in that codon wheel practice, make sure that you guys get that in by noon to the Google Classroom assignment site on that. I will be putting up a key for that, so I'm going to grade them on there, but I want you guys to have a key for that as well. That will be going up at noon, so if you hand it in afternoon, it's going to be marked late. Also, to let you know, I will be available. I'm going to be doing a Google Meet with you guys. For anybody who is interested, it's not mandatory, but I'll be sending that code out close to 11:30. I'm going to do 11:30 meet on Wednesday. I'm going to do one for 9:30 on Thursday. So I'll send that code out about 5-10 minutes ahead of time. I'll stream it right onto the Google Classroom site. That way, if you guys have questions and we need to kind of chat face-to-face, or need to go through anything, those are the two times. Neither one is mandatory, but you know, if you need to ask some questions, that's a great time. Or you can just stream them to me and I'll make sure that I get them as well. So what we're looking at today, yesterday I had sent to you guys on a video that I kind of wanted you guys to go through this, and we went through the back a little bit. I want to give you guys the answers so that you have them and just kind of run through that whole formation of protein synthesis once again, because it goes through it step by step. So on the front page of that RNA formation sheet, you guys saw a couple of things. What you should realize, and I said you didn't have to go through and color them, but it's a great little key so you guys know what you're looking at along the way. So it says, okay, well this piece here, right here is number 5. There's my chromosome. If I break it down a little smaller, double-stranded DNA. If I look at this little segment here, it's a gene, and what I'm doing is I'm transcribing that DNA, that little section of DNA, into messenger RNA. And so that little messenger RNA is this little strip right there. And they're just kind of taking that, blowing it up, and that's that same messenger RNA that I'm looking at there. I know that it's messenger RNA because I have uracil in it, and it's also a single strand. So this whole front page that you guys are looking at is that process of transcription. Okay, so if you're looking at some of those questions, what they're asking you, are both strands of DNA needed to make RNA? No, I mean, we can make RNA off that single strand, so I don't actually need that one side. We're transcribing a gene. I can just use the one side there. Does RNA formation occur in time along the entire length? No, we're just doing a small section of it. I don't need to transcribe the entire chromosome. I need a small section, and that is because that section is known as a gene, which is number 3. Number 4 says, how are these nucleotides different? Remember, a nucleotide is a sugar, a phosphate, and a nitrogen base. My sugars are different. RNA has ribose, DNA has deoxyribose. And one of my base pairs, RNA has uracil that replaces thymine in DNA. It says adenine in DNA always pairs with, it's going to be uracil when I get to that RNA nucleotide. So A will go with U when we get to messenger RNA as well as transfer RNA. What type of RNA is complementary to DNA? It's going to be complementary to messenger RNA. That is what we're making right there at that particular section. Next page, you guys are now looking at the process of translation. We're taking messenger RNA and now we're creating a protein. This whole thing is now going to occur out in the cytoplasm. We've taken that single strand that we have there. So as we go through, couple things. These parts here that you guys see at the top are going to be my amino acids, 20 different ones. They will be connected by number 2 here, which is going to be that peptide bond. So as this transfer RNA that you see here kind of actually looks like a little T, it has an anti-codon, 3 base pairs on the bottom, is going to come in and find its complementary codon. It will form another peptide bond to the amino acid before it. This big pink structure that you have, that is number 3, that's going to be my ribosome. Our RNA is what's joining them together and activating them. Messenger RNA came into the ribosome, and he's kind of moving through in this direction that we see here. He will be read 5' to 3' end. So it's my 5' end that's going to enter in first. This first codon, 3 base pairs on messenger RNA, was read. It matched to my anti-codon, so he was here. He already left. He left behind his amino acid. The next one comes in, codon, anti-codon, they're complementary together, meaning C goes with G, C goes with G, G goes with C. Bring in his amino acid, peptide bond. Next one comes in, complementary codon, anti-codon, next one joins together. So it says how many messenger RNA bases are needed for one amino acid. Each codon, each 3 bases are going to code for an amino acid. Remember, it's the messenger RNA code that's coding for the amino acid. And that messenger RNA code originally came from my DNA. The messenger RNA codon is complementary to the anti-codon in transfer RNA. It says a protein containing 100 amino acids. What is the minimum number of messenger RNA bases that are needed? So there's actually one of two ways that you can go here. We could say, oh, well, if I'm looking at 100 amino acids, it would be 300 because every 3 base pairs. But if you guys, I would take that answer. I would take 303 because the last codon probably is going to be that stop codon that does not code for a particular amino acid. So either 300 or 303, depending upon how you guys are looking at that particular question. How does transfer RNA correctly read or translate the messenger RNA? Well, all of this is done at the ribosome, and the ribosome is kind of going through and reading that particular code, making sure that that correct transfer RNA comes in. And remember, it's getting read 5 to 3 prime. The sequence of messenger RNA bases determines the sequence of amino acids in a protein. If I change one base pair, so instead of an A, maybe I have a C here, is that going to change the corresponding amino acid? Well, absolutely could, not 100% always, because amino acids are coded for by more than one codon, but it could make a completely different amino acid, which is going to give me a completely different protein. So yeah, that could absolutely happen. If some amino acids are not present in the cell when protein synthesis is occurring, will the protein be completed? No. If you don't have it there, there's nothing for that amino acid to come in. And that we talked about in the beginning of the year, the importance of eating a balanced diet. So next page, you guys should see everything there. So we have everything going on, the entire cell, the nucleus, you have transcription, you have a little RNA editing that we talked about, getting rid of those introns and keeping the exons. Exons get spliced together. That's my final messenger RNA code. My ribosome is going down, reading that messenger RNA strand. Transfer RNA is coming in with the attached amino acids. Amino acids are floating around out in the cytoplasm. So it says, which RNA performs the following functions? We know that transfer RNA is going to transport the amino acids to the assembly site, which is where the ribosomes are. Messenger RNA contains the blueprint for protein synthesis. Absolutely, because he got it from DNA. rRNA is the site for protein assembly. It's that ribosome rRNA is putting together. Messenger RNA formation occurs in the nucleus. That process is transcription. We know that messenger RNA is read at the ribosome. That process is called translation. Messenger RNA leaves the nucleus through the nuclear pores. There are actually little openings here that DNA is too large to get through. Amino acids are transported to the ribosome by transfer RNA. So you can actually see they're kind of picking them up together here, and they're going to get transported there. Each transfer RNA carries a specific amino acid. Since there are 20 different amino acids, the minimum number of transfer RNA needed for protein synthesis is actually 20. However, remember, there are 64 codons in messenger RNA. There are 64 different ways that I can arrange A, U, C, and G in groups of 3. However, there are only 61 anticodons in transfer RNA. And the reason is because of those 3 stop codons that we have in messenger RNA. They do not code for an amino acid, which means that there's no transfer RNA that is going to be used to pick them up. The back page, I'm going to kind of let you guys go through it. I'm just going to kind of explain, but you guys can kind of pause, check out your answers, make sure that they're all correct. So once again, I said that the bottom line here was your amino acids. These are the codons that are associated with it. So we said that some codons, like leucine, have more than one. Most of them are going to have more than one codon that's coded for it. You guys will always use the first line if you're given the amino acid first. So methionine, alanine, so on, these are all my amino acids. They are all joined by those peptide bonds. They are brought in by my transfer RNAs that have my anticodons on them. My anticodons on transfer RNA are complementary to my 3 base pairs on messenger RNA, which are those codons there. My codons, at the same time, are complementary to my DNA strand, which is inside the nucleus. So AUG on messenger RNA is complementary to TAC, my triplet in DNA. So T goes with A goes with U, C goes with G. Okay, so that's kind of the entire process that you have. Pause that video, check out your answers. If you guys do have questions, Google Meet, like I said, 11:30 today, 9:30 tomorrow. In a moment, you guys will kind of see a second video. For that second video, sheets that you guys are going to need. You guys are going to need a sheet within your pile. You should have one that looks like this. Okay, so you're going to pull that out. We're going to go through that one together. The second sheet that you'll see is kind of the DNA review sheet. It is double-sided. That's going to be more for you guys to go through and complete on your own and ask questions as you have them. Okay, so we'll be back shortly with that second part of it. Thanks.